Well, thanks, Guy, and thanks to the other organizers of this meeting and those folks who stick around to hear what we have to say. Um, this slide kind of serves as my introduction because we're going to talk a little bit about bull fertility, but we're going to talk about some new measures of bull fertility and then talk about some of the nutritional effects. And as Zach mentioned, I am going to share some of the data with you that Carl Dolan and we have produced. This slide also kind of mimics what my mind kind of looks like going to work most mornings. Kind of cloudy and foggy, and maybe don't know what we're getting into. And hopefully by the end of this talk, we've cleared things up for most of you. So with that, I want to begin with the role of bull fertility in herd fertility. And I know that most of you know this, but 87.5% of the genetic makeup of the calf crop is determined by the last 3 bulls used. So if we have any ability to improve herd fertility through bull selection, we need to put some emphasis on selecting bulls that are of greater fertility. I know that we talk about reproductive traits being less heritable, but I think a big part of that also comes from the fact that reproductive traits are extremely difficult to measure. So I think that's a big part of that. In addition to the role of the bull in herd fertility, the role of the bull in individual breeding season fertility is obviously extremely important. I can hear Dr. Dick Sackey saying that the most accurate test of a bull's fertility is obtained by using him to breed a sufficient number of females that are known to be of normal fertility. And it's exactly right. Of course, we can't do that for every bull because the economic outcome could be disastrous for those bulls of low fertility. And by the time we determine the bull's fertility, it might be a little bit too late. So one of the things that is important is that we realize the fact, and there's a lot of data to suggest this. This was an easy set of data to share with you, that there's a lot of variation between bulls as far as bull fertility goes. And these bulls are even bulls that were collected by a commercial AI stud. So they already have met a minimum amount of fertility, if you will. If you look at AI pregnancy rate across the top, bulls A and B had the greater pregnancy rates, and none of the measures, the single measures that are listed here, were measures that they were highest in. So the odds of us identifying a single measure for a bull, for bulls as far as measuring fertility is probably pretty slim as well. I don't know how many people know this, but it's because of the severe winter in 1949 in the Rocky Mountain region, which does predate my life, surprisingly to some of you. Scientists and veterinarians developed a bull breeding soundness exam, and I know that our next speaker's going to talk about this a lot. This is what that exam looks like, and I only want to drive home 2 points. One of them is that the goal of the breeding soundness exam is really to identify the subfertile bulls. It doesn't really help us in identifying more fertile bulls. It is also a pass-fail exam, and if we're going to make any improvement in herd fertility through selection, we're not going to make it with a pass-fail exam. A couple other things I want to point out, then I won't steal any more of Dr. Perry's presentation, is that the minimum requirements for a breeding soundness exam, the minimum motility is 30%. And I don't know how many of you passed a class when you got a 30% in that class, but it's pretty rare. And so I think that our minimum requirements for a breeding soundness evaluation need to be elevated a little bit. The Canadian breeding soundness exam has a minimum motility score of 60%. The 70% normal cells, as far as morphology goes, is a fair rating, but these are also very subjective measures. And so what I see when I look in a microscope and what somebody else sees when they look in a microscope could be 2 different things. And so I hope I don't upset a lot of folks here, but veterinarians that pass subfertile bulls are not doing us any favors. And I'll come back with this comment a little bit later in my presentation, but we really need to identify veterinarians to come test our bulls that are actually critical. And I was impressed with Dr. McFarland's presentation that 45% of producers in California conduct an annual breeding soundness exam, because I would bet that it's not that high in Montana. Some of the recent advances and improvements in bull fertility and bull sperm fertility assessment is where we're going next. If I had any talent at all, a couple of the moderators' faces would probably be painted on these two sperm that are shown on the right side of your screen, but I don't have that talent. So let's move on. One of them is computer-assisted semen analysis, CASA. Essentially, it has about 12 or maybe more measures of motility. That's wonderful, because we know motility is pretty important in the fertility of a bull, and it's extremely valuable in research setting, but it requires a microscope and a computer, so it's not going to be very user-friendly for the veterinarian that's testing bulls chute side. An iSperm has been developed. I have to wonder about the individuals who are looking at their iPad and come up with the idea that, hey, I could use this to evaluate sperm fertility. It also is able to evaluate motility. The important thing here is it's objective, just like the CASA. It's portable and it is simple. It also comes up with a concentration measure. You have to dilute the semen before you run it through the machine, the iPad, or before you show it to the iPad, I should say. But it's pretty simple to use, and it has some potential, but I think it needs a few improvements moving forward. But I commend whoever did have that kind of a mind to come up with that idea. It wouldn't be somebody like me. What we're using a lot of, and I want to talk about next and probably most, is a flow cytometer. The flow cytometer we use is shown here. It's a Guava Easy Sight. And the beauty of the flow cytometer is there are already kits available for measuring viability, acrosome integrity, mitochondrial potential as a means of measuring energy potential of sperm, DNA integrity, and oxidative radicals, reactive oxygen species, kind of as an indicator of the stress susceptibility of sperm, or the ability of the sperm to withstand stress might be a better way to say it. But sperm can be measured with a lot of other assays, such as ubiquitin assay. Peter Satovsky, Carl Kearns there. Peter's lab developed the ubiquitin, the POP assay, the post-acrosomal WW signaling protein assay that's involved with measures of oocyte activation or the ability to activate an oocyte. Zinc signature or capacitation assay that Carl has worked on so much. And there's other novel biomarkers, and we've got some people who are working on some of those as well. I know other labs are using flow cytometers, and I think it has a lot of potential. I think all speakers would agree that we can't make a ton of progress with a pass-fail exam, so we need to come up with some better ways of measuring fertility. And so the flow cytometer is pretty valuable. The way this works is we have to collect semen sample, mix it with different fluorescent stains for the different markers of interest. We run it through a flow cytometer, which essentially has the sperm pass by a laser single file. That laser is detected then by a forward scatter and a side scatter detector, which are the blue ones on this screen, and then different colored detectors for the different fluorescent stains or dyes. And then it comes through a computer and spits out a picture that looks like this. And the person running the machine has to identify the cells that they're interested in. So they draw a circle or a parallelogram around the cells of interest, the sperm cells, because they're separated really just by size and density on the first plot. And then it can actually be programmed to measure different things. So we can measure live and dead sperm and other variables. There are more expensive flow cytometers than what we use. An image stream is shown here. It's kind of like a flow cytometer and microscope in one box. So it's showing you the actual sperm it's evaluating with bright field and then different dyes that you've added to the reaction. So it's pretty valuable in that respect, that you can see what it is that it's actually evaluating. There's even some handheld flow cytometers that might potentially be a future veterinarian tool if we can identify which of these measures are more valuable than others. The current handheld flow cytometers are about $10,000 or $12,000, and they already can do cell counts and viability and things like apoptosis. They are expandable. You just need to program to do them to measure other things. Back to the flow cytometer. We need to use the fluorescent imaging with a microscope because we don't have that fancy one, but I want to give everybody kind of an idea of what some of these things look like. With the microscope, a live versus dead sperm. The acrosome cap on the head of the sperm is indicative of acrosome damage, and you can see it in the above picture as well. The sperm that are red are dead sperm, but they've got a bright green cap. And then in the mitochondrial membrane potential assay, for example, a polarized sperm versus a depolarized sperm that is indicative of its ability to continuously or for a long period of time at least produce energy. The acrosome integrity and viability, when we run this assay on cells, the first thing we do, as I indicated, is we get a forward scatter and a side scatter screen that or plot, and we have to put a parallelogram around that which we're calling sperm. Some of the rest of this is debris from the ejaculate or from the straw, and then it sorts it, in this assay at least, by yellow fluorescence and red fluorescence. And then we tell again to use this as being the portion that is actually sperm that we're interested in evaluating. And it'll break it down into live and disrupted, dead and disrupted, live and intact, and dead and intact. And it actually produces a table that shows that the lower left quadrant of this last plot is the live intact sperm, and that's the sperm that we're interested in. With the mitochondrial membrane potential, as I indicated, the first plot is a forward scatter, side scatter again. Second one is orange versus green. And the third one is also orange versus green. But we've eliminated some of the debris again. And as the sperm are polarized with the JC1 dye, they move upward as more orange as opposed to green as they become depolarized. Okay, so the problem that we have at the moment is that we have insufficient field fertility data to identify which of these is the most valuable fertility biomarkers. One of the things I didn't say about the flow cytometer is this flow cytometer is also capable of assessing up to 10,000 sperm in a minute. That's way, way, way faster and to a much greater degree than we're able to look at through a microscope and count across a field of view, for example. So what has me convinced, though, that this is a way of moving forward as far as measuring bull fertility or confidence in some of these measures I'm going to share with you in a story. We were trying to work with a very large producer to help us develop some better markers or identify better markers of semen and bull fertility. And so we wanted to have them conduct breeding soundness exams, send us ejaculates. They were already genotyping calves. We would be able to identify which calves came out of the breeding season from which bull and correlate that maybe to some of the measures that we had on those bulls from that semen assessment. And they said, well, why should we work with you? What, can you convince us that what you're doing has any value? And I said, well, I'm not sure we can. But I asked them if they had any historical field fertility data on any bulls that they've been using. And they did. They had semen banked on some of those bulls. And I said, send us some semen on some of those bulls that you have field fertility data. We'll analyze it every way we know how and see if we can convince you that some of what we're looking at has some value. So they sent us about 30 straws from about 30 sires, and called about a week later and said, "What have you learned?" And I said, "We haven't learned anything yet. We're trying to determine where to start with this." I said, "Can you identify maybe 8 bulls that we could start with?" And that was what we were able to do in a day for these measures. And so they did. They picked 8 bulls and said, "Start with these. These are the highest priority bulls. Some we're still using or want to use, and some we haven't anymore." And so we ran them through a repertoire of flow cytometry exams, assays, and came up with a composite score, which is just really an addition of the ranking of each of those flow cytometry measures, and gave them a composite fertility rank, which is on the left side of the screen here, of 1 through 8. And I thought, before we share this data with them, that's only one shot to convince them that we have some value in what we're doing. So we also coded them a little bit more as being high, moderate, or low fertility. Sent the data to the producer, and somebody called us within about 30 minutes and said, "Yeah, we're interested in working with you because you did identify the highest fertility bulls and the lowest fertility bulls. I think 3 and 4 or 4 and 5 were switched a little bit as far as field fertility, but they really didn't differ at all." So they were enthused enough to continue or to start working with us. And so we're excited about some of the opportunities that exist there. And we haven't gotten much further off the ground as far as that study has gone, or is planning to go. Okay. So, but this gives me some confidence in some of these measures. So Dr. McFarland also mentioned bull care during the off season. And if the Californians are anything like the Montanans, the bulls at your operation get the poorest pasture, the poorest quality feed, and yet we expect more of them than we do an individual female when it comes breeding season. So it's a little bit surprising. And so we thought, well, the easy cherry picking studies that we can do is some nutritional studies to determine how this care during the off season is influencing the fertility of bulls moving forward to the next breeding season. So we started with a study of the effects of mineral source on mature bulls and sperm characteristics. We had some preliminary data that suggests that different minerals played some role, but never had enough data to say it was really convincing or how it was improving fertility. I'm not sure we've got enough data yet, but we started with 37 bulls. They were different breeds, and so they were blocked by breed when we assigned them to treatments. But the treatments are shown here. The majority of these bulls had absolutely no mineral supplement provided to them for 2 months prior to the onset of the study. And 24 days before the onset of the study, we collected a liver biopsy from these bulls and then used liver copper concentration as a means of again using a block to separate bulls into the different treatments. Our treatments began on day 0 and were fed for 71 days. Consisted of no mineral supplement for the continuation of that period. Sulfated minerals, copper, zinc, and manganese sulfate, or the chloride-based minerals, basic copper chloride, zinc, and manganese hydroxychloride that comes from Intellibond, the micronutrients minerals. There were 12 to 13 bulls per treatment. We did have a mid-test semen collection, blood and weight collection on these bulls, and then we conducted a liver biopsy again at the end of the study on these bulls. The results of these bulls, of this study rather, not very exciting, honestly. The bulls provided no mineral, or the sulfate mineral were liver deficient in copper on day 71. The bulls provided the chloride mineral were not mineral deficient. Bulls provided the chloride mineral tended to have greater sperm concentration and progressive motility in the day 71 ejaculates, but that was about it. Not much in our flow cytometry measures that we saw anything different between treatments. When we looked at correlations, liver zinc concentration was correlated inversely with sperm acrosome damage. So bulls that had higher levels of zinc had less acrosome damaged sperm in their ejaculates, and a greater liver concentration of zinc. Again, bulls produce more sperm in the ejaculate, or a higher concentration of sperm in the ejaculate. That was about it. So we said, well, we're going to look at peripubertal bulls, and so we decided to look at the effects of copper and zinc primarily during yearling bull development on sperm fertility. We used an 84-day trial that included 124 peripubertal bulls, 4 treatments. The next slide. We conducted liver biopsies at the beginning and end of the study. And in the second year of the study, we also evaluated the semen for mineral concentration. These bulls were fed in Kaelin gates, individually fed, and from weaning until the start of this study, day 0, again, these bulls received no mineral supplementation as they were getting bunk broke and beginning to utilize a feedlot and get used to eating in the feed bunks. Treatments were control, no copper or zinc for that 84-day period, zinc hydroxychloride or copper hydroxychloride, or both zinc and copper, zinc hydroxychloride, copper chloride, basic copper chloride. I said this was a two-year study, 31 bulls per treatment. Okay. As far as the liver copper concentrations in peripubertal bulls, supplemented with the mineral for 84 days, the y-axis is liver copper concentration in parts per million, and the x-axis is the different treatments. The orange bars represent day 0, and the blue bars represent day 84. You can see that from that the bulls that were fed copper in either copper alone or zinc plus copper had elevated concentrations of copper in their livers on day 84 compared to the control bulls and the zinc treated bulls, which were deficient or marginally deficient in copper at the end of the study. However, 2016 and 2017, the bulls didn't act the same. So in 2016, the bulls were not copper deficient on day 0. And throughout the period from day 0 to day 84, copper concentration decreased, or at least on day 84, it was decreased compared to day 0 across all treatments. The bulls that received the copper alone did have greater concentrations of copper in the first year. In 2017, the bulls were copper deficient on day 0, and only those bulls that received copper in their supplements were copper adequate as far as liver concentrations were concerned on day 84. How that affects the semen. Again, the left graph shows the liver concentrations that I showed you in the previous slide for 2017. The graph on the right shows you the semen concentrations of copper for the different mineral treatments. Change in liver copper concentration on day 84 did not impact semen copper concentration. So even though it was increased in the bulls that received copper for the liver, it wasn't different between treatments on day 0 or day 84 in the semen of those bulls. As far as the liver zinc concentration in those bulls was concerned, liver zinc was increased in bulls fed minerals for the day 84 for the 84 days. And by this, it's a comparison of mineral versus control, is why they have the A, B, and B across the top there. Not the dramatic differences in liver concentrations of zinc between 2016 and 17. When we compare the liver concentrations of zinc with the semen concentrations of zinc, there were no treatment differences in semen concentrations of zinc on day 84, day 0 or day 84. And the liver and semen concentration of zinc did increase from day 0 to day 84. As far as the standard breeding soundness evaluation measurements, there were no treatment effects, but from day 0 to day 84, each of the measures that make up portions of or that we evaluated on these bulls, scrotal circumference, ejaculate volume, concentration of sperm, motility, normal morphology, viability, all improved from day 0 to day 84, just as you would expect. The percentage of bulls that passed the breeding soundness exam from day 0 to day 84, when bulls were roughly 13 months of age at day 84 in this study, all improved. Okay, so we know that this is improving generally across bulls, and it did again in this treatment. Again, that comment about veterinarians that pass subfertile yearling bulls because they believe their fertility will improve as the bulls get older are not doing producers any favors, because a number of those bulls don't improve. As far as the flow cytometry measures go, again, we looked at acrosome integrity, mitochondrial energy potential, DNA integrity, viability, 3-hour viability, reactive oxygen species. They improved in each of those bulls from day 0 to day 84, but there were no differences between mineral treatments. Again, that kind of surprised us. So in conclusion, on mineral supplementation in bulls, zinc and the copper supplementation had only very minor improvements in sperm morphology. I didn't talk about that. Percent normal spermatozoa in the ejaculate did not differ, but some of the abnormalities differed slightly. But again, we're talking 3 to 6%, so it's pretty minor. And so I think that bulls have very low requirements for zinc and copper, and or they're very efficient or have very efficient homeostasis mechanisms for recycling and or conserving zinc and copper. One note I have to make after, especially after seeing the previous two speakers' presentations, is that these were bulls collected every 28 days and may not reflect the mineral needs of bulls during a breeding season or bulls that are developed where mineral antagonists exist. So the presentation or the set of data that Zach referred to on differing planes of nutrition effects on mature bull fertility with Carl Dolan at North Dakota State University. As Zach introduced that topic, there were bulls on a positive energy balance or negative energy balance in year 1. And then those same bulls were brought to a common point. And the next year, those that were on the negative energy balance previously were put on a positive energy balance. And those that were on the positive energy balance in year 1 received the negative energy balance. There were 15 bulls in the study. Semen was evaluated every 28 days. And on day 112, which was the end of the study, semen was frozen, and that's where we came in the picture and started evaluating some of that semen. Now, I want to share with you this slide, which is the body condition score of those bulls from day 0 to day 112. Zach referred to this a little bit. Really, by day 28, those bulls were starting to spread apart and different in body condition scores. These are the post-thaw flow cytometry measures for the plain and nutrition bulls. Proportion or percentage of sperm on the y-axis. Positive energy balance is in orange, negative energy balance is in blue. As far as the viability went alone, no differences, although the blue, we kind of were thinking that those bulls on a negative plane of nutrition would have a little bit lower viability. And so it wasn't statistically different, but it's going in the direction we anticipated. As far as percent live and intact acrosome, identical. As far as mitochondrial energy potential, this one kind of surprised us. The bulls on the negative energy balance had much greater energy potential in their sperm. And as far as the reactive oxygen species, better able to withstand stressful situations. Capacitation, acrosome reaction, those are all things that produce oxidative radicals. And so their better off from that standpoint if they were on the negative energy balance. I think that the take-home message from this is probably that it's related to body condition scores in these bulls. And if you look at the body condition score on day 12, those bulls on the positive plane of nutrition were almost a body condition score 7. And as David and Zach both referred to, scrotal temperature regulation probably is messed up when we get to bulls to a body condition score of 7. Those bulls at a body condition score of 4.2 on the negative energy balance, they were not messed up as far as a temperature regulation, but certainly scrotal circumference was lower in those bulls at that time. But motility was not different between the bulls on the fresh ejaculates throughout this study. So another study that we conducted with Carl Dahlin was looking at the effects of dried corn distiller's grain plus solubles, DDGS, which is high in sulfur, or a diet that had equivalent sulfur on peripubertal bull fertility. This included 36 half-sib Angus bulls in this study that had been weaned a couple of months prior. The weights are shown there as well. Diets were a corn-based diet containing approximately 60% concentrate. It contained about 0.2% sulfur. Diets containing 60% DDGS as a replacement for corn, containing 0.54% sulfur. And then the control corn diet that had an equivalent sulfur added as the DDGS diet in the form of calcium sulfate. Bulls were individually fed in Kaelin gates. Kind of the treatments and timelines are shown here. On day 0, bulls were weighed, and they were weighed every 14 days. And the amount of feed delivered to those bulls was adjusted to attain an average daily gain of about 3.5 pounds a day. At day 112, semen was extended and froze and sent to us, and that's where we came into it again. This graph shows the post-thaw flow cytometry measures on these bulls. Again, percentage of sperm on the left axis. Orange bars represent control bulls. Blue are the 60 DDGS, 60% DDGS bulls, and then the light green bars represent the bulls that had the control diet but added calcium sulfate in their diet. If we look at viability, no differences. Live and intact acrosomes, no differences. When we look at the energy potential of those bulls, the bulls fed the corn-based diet with increased sulfur, we see increased energy potential. As far as the reactive oxygen species, no different. DNA fragmentation, no different. And so the take-home message on these studies as far as nutrition and bull fertility goes is that based on the studies that we've conducted in bulls, and I have to qualify this again, this is bulls outside of the breeding season, we've not found major effects of nutrition on semen fertility measures. The two possible things that we have seen is that overfeeding may be what's producing the greatest negative effects as far as the sperm energy potential. Certainly scrotal circumference was less in those bulls on the negative energy balance plane of nutrition. Stress susceptibility of the sperm was greater in bulls that were overfed. And then bulls that have greater liver zinc concentrations or have correlated with producing ejaculates with less sperm acrosome damage and greater sperm concentration. So about to the end of my presentation, and I want to wrap this up talking about future semen measurements and how we're going to make this all work. And it's going to take a team effort because we certainly don't have all the answers. But these are the semen measurements that we can pretty easily measure on bulls. And they include the volume of the ejaculate, concentrate, motility values, whether it be with a CASA or not. CASA would be better. normal morphology, acrosome integrity, mitochondrial potential, DNA integrity, these other measures as well. I think the key to identifying semen that differs in fertility requires identifying the value of each of these measurements to produce a bull fertility index. And I would love to see the industry go that way. But like I said, it's going to take a lot of team players working together on this effort. I don't know what these factors would be that you'd measure, that you'd multiply by motility and normal morphology and acrosome integrity and mitochondrial potential DNA fragmentation index. I don't know what all those would be and then what other markers or biomarkers we might look at, but I think that's the way we have to go if we're going to make any improvements in assessments of semen fertility. And then we still have another big door to go through to go from semen fertility to bull fertility, because the semen measurements that we're making generally are on frozen thawed semen as opposed to fresh ejaculates. So that's a pretty important component of this moving forward as well. So the question becomes, can a bull fertility index provide a quantitative assessment of bulls or semen to allow selection for fertility? And I believe it can. We just have to identify the components of that. So with that, I'm going to thank you, but I too need to acknowledge some people, and especially the first person on this list who does the majority of the work that I share with you folks, and really is the guts and the brains. I'll even give her credit for that, behind all of what we're doing here. So I appreciate working with all these other collaborators as well. And with that, I'll open it up and I'll answer questions to the best of my ability.
All right. Thank you a lot, Tom, for that excellent presentation. Have a number of questions coming in, so please put those questions into the Q&A box. First question is, in regards to the data that you showed, is there any data that you've looked at comparing the flow to, let's say, other functional assays, something like a swim-up assay or mimicking the inside of the uterus, something like that, cow's heat versus non-heat, along those lines?
We have not done that yet. We've talked about doing it relative to sperm swim-up, and even doing sperm swim-up first, and then evaluating that fraction that made it through that and the fraction that didn't. We just haven't got there yet.
And also, yeah, also you mentioned this a little bit, a lot of the work is done in regards to frozen thawed sperm. I think that's what a lot of us have done things on. And so just wondering what your thoughts are in regards to, how do you get a good measure of field fertility? We can probably get that pretty easily, if we look at number of inseminations and that on the frozen thawed side. What are your ideas there on the natural service side? What's the number of calves that need to be born to sort of validate that? Just any thoughts you might have around that, and maybe tips for producers on collecting that data in a way that will end up being able to help us as researchers develop those tests.
Sure, and I think this needs to begin with evaluating semen first, and then maybe we can go to the bull after that, because as you know, we can inseminate a much larger group of females with semen from different bulls. And I'm got to give Mel DeJarnett credit for my answer here. Is it's going to take thousands of observations on a individual bull to get a comfortable level of what his fertility level is, high, low, or average, as far as identifying some of these traits. And the semen collection studs don't generally produce much semen from bulls that would rank or fall into that low fertility group. And so we really need to work with them to say, look, if you've got some bulls that you don't think are freezing well, or you think the fertility is compromised, let us have access to some of that so that we can evaluate it and see what we think as well. When it comes to natural service, hopefully we have some of those answers so we know what some of the targets are we're looking at. And then maybe when we maybe we can program some of these small handheld flow cytometers to evaluate those specific parameters primarily.
Excellent. So could you just expand a little bit on maybe how a BFI would work? Are you thinking it's a sort of an individual semen analysis or something that's similar to an EPD based on the results of relatives, progeny, etc.? And just expand on that a little bit.
Sure. It would have to be somewhat similar to an EPD, I'm sure. But, you know, like sire conception rate or some things like that, we have plus 4, plus 10 maybe, and then negative values as well, just different from the average. And I know the average is probably going to change as time goes on as well, if we're able to make some improvements in fertility. So I think it would have to be a function, something like that. But I do think that it's possible. And in fact, I think if it came to using a handheld flow cytometer to help with the fertility exam shoot side, for example, I think those could be even programmed to come up with a fertility, bull fertility index itself. So I think that possibility exists.
Great. In regards to, so the study that you referred to at North Dakota State, those were all mature bulls, correct? If I remember correctly.
Yes.
Okay. So
the diverse plane of nutrition bulls were mature bulls.
Yeah, sure.
Yep.
So there's a question here in regards to when you look at, if you take 100 bulls that are determined to pass a BSC at 18 months of age with a 70% or greater morphology and 30% motility, how many of those bulls will fall out, or what do you think the fallout rate is by 5 or 6 years of age because they fail a BSC? So if you take nutrition and other factors out, I know that's a really tough question, but just kind of some thoughts there.
Sure. So it might be different across the country. Certainly probably is, because we have bulls that experience frostbite in the bottom of the scrotum. And so that might be the biggest reason we have problems here in Montana. But we have seen pretty consistently 10 to 30% of the mature bulls each year fall out. So honestly, by 6 years of age, there aren't always a lot of bulls that are still bulls that we would continue to be using.
Excellent. And the last question that I'm going to ask here before we move on to one of the award presentations is, if you were advising a producer in regards to whether you feed your bulls during the breeding season, just that idea of supplementing your bulls specifically during the breeding season, and is that going to help a lot, do you think? Is it something that's not going to help a lot? Just kind of what your ideas are in regards to that, and what you talked a little bit about on maybe some of the nutritional aspects that y'all saw into very limited changes in semen morphology.
Yeah, I don't know how you would do that to begin with, Kai, because I think bulls that are worth their weight at least are the bulls that aren't very interested in what extra feed you're providing them. It's the bulls that are out trying to breed females. And so I think it would be tough to provide a supplement specifically for the bulls and improve their abilities in that regard. We have not evaluated fertility of bulls at the end of a breeding season nearly to the level that we should be, to see if the bulls flow cytometry measures, for example, are similar at the beginning of the breeding season and at the end of the breeding season. But it's certainly something worth doing.
Excellent. Well, Tom, we really appreciate you joining us today for this presentation and taking time out of your schedule. There are 2 questions left in the Q&A. If you have time, if you don't mind trying to answer those. Again, we really